Each of the centers have proposed basically a speaker to actually give us an insight into some of the aspects of the different work that is happening around. And what we've all heard from John is that Dean is going to tell us about a journey that took him all the way from basic research to quite close to basically even commercial enterprises.
Yeah, great. Yeah, thanks very much. So the title of the talk today is From Neural Fields to the Clinical Field in Epilepsy. And the reason why I chose this talk is because a lot of my earlier work was motivated by the work of John Terry in neural field modelling, and of course Wes and Mark Goodfellow down the back there. And that work is really cutting edge stuff, and I just first of all like to start off by thanking these guys, not just because of their contributions to the field, but also for being thought leaders and helping our team with publications, but also with very rich discussions with numerous trips backwards and forwards, and it's been extremely productive and certainly guided my journey over a course of about 10 years from taking this work from basic research through to clinical application at a relatively large scale. I'd also like to thank, right from the very get-go, Pip Caroli and Mark Cook, who's here today. So if you guys get the opportunity to talk to these guys, please take the opportunity, as well as myself, because a large part of the work is done by these guys, of course, and of course many other people. I'd like to just make a couple of disclosures. First of all, I'm going to talk a lot about SEER Medical, which I'm the CEO and co-founder and have a financial interest. But also, I'd just like to ask you to please not take photos or videos, because I'm going to show some images from patients here who have given us permission to show these here, but we don't want them to end up on the internet or circulate any further. So if you could do that, that'd be great. So I've got about half an hour here today to take you through this research, and it's really split into three parts. So first of all, I'd like to give a bit of a background about epilepsy and some of the problems that we face and some of the interesting engineering problems that we have the chance to tackle in the field. And then I'd like to talk a bit about some of the long-term devices and implants that we've been using in Melbourne to try to get to the bottom of some of these challenges. And then finally, I'd like to talk about some of the commercial aspects that we've sort of moved on to now, which is pretty exciting. Okay, so some introduction. So we work in the field of epilepsy, and epilepsy is a relatively large problem. It affects around 1 to 2% of the world's population, so it's about 75 million people worldwide, and about 25 million people don't have sufficient treatment where they can become seizure-free. So that's about equivalent to the population of Australia, just take that into context. So we believe it's a really big problem. Now, seizures, we don't see them so often because seizures are very debilitating and it often restricts people's day-to-day life. And although they can be very rare events, they can carry significant risk of death. So if seizures become bigger and more and longer and more frequent, there is this chance where patients do not recover, unfortunately, and they have an equivalent impact as the road toll in our home state as Victoria, and so it's a really big problem that often goes underlooked. Now, the mainstay of treatment is drug therapies, and of course it's been a very rich area of development. And I was over in Washington DC on the way over here with the meetings with FDA around ways to construct new clinical trials, and the FDA were talking about the great successes that they've had over recent years with lots of drugs being approved. But when we really dig into this a little bit deeper, we can see since about 160 years ago, the rate of seizure freedom with bromide from GAL was about 60%, and now we've moved up to around 70% seizure freedom. So although there's a lot of drugs available now, there's still a major problem in terms of treatment. And so we believe that we can fill this gap here with these patients with seizure forecasting. So although it's not therapeutic, what it does is it gives us opportunity to intervene, I guess, to provide a warning so the patients can get to a safe place or safe conditions when they do have their seizures. And I've just shown some little examples here with this patient here because although seizures may be rare, where they occur can cause major problems. So for example, if someone was swimming or if someone was driving a car, operating machinery, then of course there's significant risks associated. So a seizure forecasting system could of course alleviate a lot of those risks by providing a warning in advance. Now I'm going to show you some examples of what seizures look like in the context of EEG. So we use EEG as a diagnostic measure for epilepsy, recording electrical fields of the brain. And we see one of our patients here at CU Medical up on the screen here, and this is an example of their EEG, their brain waves, and I'm just going to show you this video here. If this will work. Okay, let's just try again. So we see the onset of the seizure is quite dramatic, and in the EEG signals we see this abrupt synchronization of the rhythms of the brain, and of course that causes motor disturbances and cognitive disturbances. And this is what we think of when we see seizures, these abrupt convulsions, and it's quite confronting. But of course there's many types of seizures out there, and this is another example here. And I've shown this one especially because our patient here that we can see just here, she's wearing the EEG system, and that's a mother there using our app from CU Medical. She's doing a rendition of the Spice Girls Stop Right Now, and so I thought you might like this being in Britain. Let's see if we can get it. And then we see the onset of the seizure.
Did you just do another one?
Okay, so I might just go back to the start of that. So the seizure onset is right here, and you see this abrupt synchronization of the rhythms of the brain. And if we watch the blue line cycle across to this point here, we'll see that there's an abrupt change in her behaviour, where she just begins to tune out.
Did you just do another one?
So it's a very appropriate example here because she's singing Stop Right Now, and then she has an absent seizure which is characterised by behavioural arrest or stopping. So it's a particularly good example. But also with the synchronisation of the rhythms of the brain, you can imagine that there's very little communication process happening, information transfer between different brain areas, which of course causes this cognitive disturbance. And so because of the nature of the relationship between the electrical fields of the brain and epilepsy, it's a really ripe area of research for engineering and mathematics with these types of signals. And I've been quite privileged to work with Mark Cook, with a lot of patients in Melbourne that have these intracranial EEG electrodes implanted for surgical purposes. So for these patients that don't have very good seizure control, the last line of treatment is surgical resection of the part of the brain that's causing their seizures. And a part of that process is we implant these grid electrodes to really map out the areas that are important in the brain that we can't resect, but also try and pinpoint down on the focus of the epilepsy. And so we have this direct electrical interface between the brain and computers. And of course, lots of engineers have been trying to take advantage of this interface by researching seizure control, by using electrical stimulation, by doing seizure forecasting, which is really trying to decode the signals of the brain, reverse engineering the brain, and of course brain mapping. And a lot of this work was really spawned on by optimism around deep brain stimulation, which is a therapeutic approach that's been vastly successful in Parkinson's disease. And of course people have been trying to catch up and chase this down for epilepsy. So seizure forecasting the field started in earnest back in the 90s, where people were really searching for a transition in the brain from chaos really to order. And the early work started with measures around correlation dimension and trying to estimate largely Lyapunov exponents in the hunt for chaos in the brain. And this is just an example here of time delay embedding of the EEG. So this is a mock of an EEG signal. It's really a Lorentz system, and we can see the Lorentz attractor here from time delay embedding. And these approaches were used with the EEG where people were trying to estimate the complexity of the system or the dimension of the system where it was thought that there was a gradual transition from highly complex to more order as patients approach seizures. And we can see the mapping here from the true attractor to the time delay embedding system here. Now, of course, that was about 10 years of work, and but then the field moved on with challenges around performing these types of analyses in terms of non-stationarity of the systems and noise in the systems, and no one could really get to the bottom of what's really happening here. And then people began to focus on estimating synchrony in the brain, which is really the same idea, where we're looking for transitions from a highly sort of disorganized state with a gradual synchronization process to a fully synchronized system. And there's numerous techniques of trying to estimate synchrony of the brain. But overall, the ideas hold across these different areas, where it's a transition from a highly complex system to a more ordered system. And certainly we were pursuing that in the early days with these types of patients here. So this is a photo from about 10 years ago, from just to show you the setup that we were working with in the lab with Mark's patients. So we had this control system here where we could directly interface with the recordings coming straight from the brain, and there was a complex array of neural stimulators and neural recording systems here. And the idea really was that we could try and detect the onset of seizures very early on, and then provide some kind of counter stimulation to try and kick the brain back out of a seizure state. And with this particular patient, we actually did this quite successfully many times here. And so we see this detection here of a seizure onset here, and then it activated the neural stimulator. This is artifact from the counter stimulation, and then the rhythms of the brain return back to normal. Now, we were able to do this in this patient here, but it wasn't reproducible across a wider set of patients. And in fact, most patients that come in for this procedure only have 1 or 2 seizures. And so in the end, it was really, although the ideas were great, it was really not scientifically well grounded in terms of being able to look for reproducibility or trying to optimize the methods involved, because there's numerous parameters and they're often patient specific. So there was a lot of challenges in that approach here. But the basic idea of counter stimulation, the theory behind it was really working around this notion of this critical transition in the brain as the brain approaches a seizure. And so the basic theory is that if this is a normal brain state here, and this is a seizure state, and a bifurcation plot here, where there's some critical parameter that's making a movement towards the left here, and when it hits K2, it makes a critical transition into a seizure state. And the idea is that you can use a counter stimulation or a stimulation to knock yourself from one attractor to the other, or one stable state to the other. And then of course there's analogies here then for seizure forecasting, in that if you could estimate where the parameter K is here, then you can measure the sensitivity to the transition to a seizure. So we wanted to be able to find measures of measuring seizure susceptibility by tracking this parameter K here. And of course, this theory of critical slowing shows us that as you move closer towards K2 from K1, that the system slows down. And that means that if you perturb the system, it's going to take a longer time to recover back to its stable state. And so in this patient here, we also tried these approaches. And so if you look a little closer to the screen here, we can see these little electrical stimulations here, which are small perturbations that we're trying to track. And so we were running some experiments here where we're trying to perturb the brain, measure its time to recover back to a steady state. And we've used this approach here in these surgical cases to really try and get to the bottom of some of these seizure forecasting approaches. And this was really the basis of our research moving forward into some of the longer term devices, which I'm going to talk about now. So that work really showed a lot of promise in the clinic, but it was very difficult to rigorously scientifically get to the bottom, a deep understanding of what's actually happening here, simply because the time duration where we could monitor these patients was too short and the seizures were too infrequent. So we needed to go back and think a bit more carefully about what was happening here. So we ran a sequence of animal experiments where we can reproduce this approach in rats, and we had some successes here. And we also did it in dogs, in canines. This was really challenging actually, because we had to learn how to do these neurosurgeries in dogs, and we also had to form collaborations with device companies, and we collaborated here with Medtronic with a human deep brain stimulation system that also has a sensing circuit involved. And so we can see our surgeons here implanting one of these DBS systems in a greyhound. Fortunately, we had a good collaboration with the veterinary school, where they have this very large colony of greyhounds which were actually ex-racing dogs. So they were dogs that were actually saved from being put down because they were too slow, and they were used as a blood bank for the veterinary school, and we're fortunate enough to be able to use some of these animals in these experiments. And I guess what we found was that the theory was holding up, and so the idea here was to do these electrical stimuli, and we can see an example of this here. We're getting this evoked potential, and the idea is that if we can deliver a therapy to the brain, then this evoked potential will be moving to this peak here will be moving to the left for you guys. And so what I've shown here with each of these dots is the time it takes to reach this peak here, and it's moving to the left when we, at this point here, we're starting to administer anti-epileptic drugs. And so there's this slow titration, but we're giving the drugs a couple of times a day to these animals, and we can see that they're able to influence the excitability, the susceptibility, and measure it using the drugs and this evoked potential regime. So this was really promising, and this led us to the next phase of the study, which was moving back to humans again. And the aim of the trial here was to try and optimize DBS therapy. Now, DBS in epilepsy is very different from DBS in Parkinson's disease, because the symptoms of epilepsy are very intermittent, whereas in Parkinson's they're always on. You always know what to do with your stimulation system. It's much easier to optimize it. But with intermittent system like in epilepsy, it becomes very challenging. And so we believe that these evoked potentials will give us a gauge on how to tune the therapy in humans. And so we went to Medtronic proposing the human trial, and they've been able to support this. And this is very early days. We've implanted one patient, and we've got another 4 to go over the next few months. But we implanted bilaterally devices in the anterior nucleus of the thalamus and in the hippocampi of these patients. And the reason why we targeted these centres is because they're a part of a neural network which is known as the circuit of Pepez. And this is a really cool closed loop where we can stimulate in the anterior nucleus and measure in the hippocampus, and then at cycles around the network. And so when we give single pulse stimuli to this network, we can excite an evoked potential that reverberates around and gives us a really good gauge on how excitable the brain is in general. And of course these devices have leads that tunnel down the neck, and they're implanted just under the clavicle in the chest. So this is a CT image here showing that the leads went into the anterior nucleus. And this is a photo from when we switched the device on for the first time in the clinic at St Vincent's in Melbourne. And so this is Katrina Dell and Matthias, who really did a lot of the work here with running this trial. Wendell D'Souza is a neurologist here, making sure everything's going okay. Emily from Medtronic. And this is the real hero of the situation here, Flora, who's very brave, and she was the first patient to be involved and run with this technique. And we can see the communication devices here on her chest that are interfacing with the device, and Kat's running the software to do the stimulation and the recording. So just a quick refresh here. So what we're trying to do is do electrical stimuli and trying to find some kind of therapy that can move us back towards this way along K1, so trying to get shorter evoked potentials. And then the way we're doing our experiments with this was to deliver these probing stimuli, these single pulse stimuli, and then provide a therapeutic stimulation, and then do another probing stimuli and look at the difference between these evoked potentials, and then repeat. And we can run this iteratively to try and find an optimal stimulation parameter set for this particular patient. And we're able to do this here. In fact, we're able to almost completely wipe out the evoked potential here when we've optimized the stimulation frequency here. So this is the pre-probing and then the post-stimulus probing, where we see a large difference in this. This is very early days, but we've been running this now for a few months with Flora, and I'm going to show you some of the data in a very unconventional way of how she's doing. So this was from our last visit into the clinic, and I got sent this photo just a couple of days ago. So we can see the data presented just here, and each of these X's are indications of days when she's been seizure-free. So it's having a dramatic effect on her epilepsy. And now she's got a very difficult to treat type of epilepsy where she has grey matter inside her ventricles. It's called nodular heterotopia. And with that comes lots of seizures. And so it was completely ruining her life. She couldn't do anything. She was stuck at home. And now she's already back at university and getting on with her life. So it's a really great story. So moving on now to another device that we've been using in the clinic. So this is a long-term monitoring device. This was a study led by Mark Cook. It's the NeuroVista study. It's been becoming quite famous, where we're able to implant 15 patients with devices. We're able to record directly from the cortex for periods of up to 3 years. So this is a really unprecedented look inside the brain over long periods of time. And so the device, again, was this implantable device. It looks like this. There was a pager component. And so this patient already had an operation in the brain, so we could put the leads back onto the surface of the brain through here, and there's a wire going down here, and the device is underneath the clavicle. CT image. You can see where the electrodes are sitting on the brain. And this device was able to record seizures continuously in a 24/7 way. But it wasn't it couldn't do electrical stimulation. So we had to look at other markers here of critical slowing in the brain, and some very simple markers, including looking at the variance and autocorrelation of the signals. And so we're able to do this in these patients, and we found some really interesting results. So I'm just going to whiz through this really quickly so we can move on to the last part of the talk. And so what we see here is the raw signals here in grey. These triangles show the occurrences of seizures, and then the autocorrelation signal here, you can see it's moving up and down at a very fast rate here in blue. There's this short cycle going on, and then there's this longer modulating rhythm in red here. And it's the interaction of these different oscillations that are occurring at these very long time scales within the brain that seem to be raising and lowering the susceptibility to seizures in these patients here. And of course this is the variance signal here. And the time scale of these long oscillations was something that was extremely surprising to us, and it's really sending ripples around the epilepsy world right now in terms of a race of trying to understand what's happening here, what's underlying these long-term cycles. And so if you look at how these influence the likelihood of seizures here, well, for starters, we can look at these circular histograms here and these Rose plots, and we can see that the seizures are distributed at this rising phase of the short cycle and also the rising phase of these long cycles, very tightly distributed in this particular exemplar patient. And if we look at across both of these cycles, so a long cycle here and a short cycle, we can see the seizure probability is really high here, and it's almost zero in this quadrant. So this gives us a beautiful signal here of being able to forecast seizures, but also try to understand the process. And of course we've used this as a seizure forecasting tool, and we can see here that, and what we've done is we've simple threshold of these signals here, and we've been able to forecast a large proportion of the seizures here, and also provide an indication the patients are going to be safe. And then of course the seizure forecasting algorithm is only going to work as if you provide risks around the times where seizures occur, because you could have a perfect forecaster if you say you're always at risk. You're never going to get it wrong. So you want to get this number down as low as possible, and we've done a pretty good job here. Now, interestingly, this very, very simple method of using this autocorrelation with understanding of these longer-term rhythms in the brain has done extremely well in comparison to other techniques, including a range of machine learning and deep learning techniques. So this is our critical slowing approach for forecasting. And in green here, it's just showing which ones are superior. You don't worry about reading this. But in just about all cases, this approach is superior to anything else, including this Kaggle contest. So we released this data to Kaggle, which is a crowdsourcing data science platform, and we had over 10,000 algorithms submitted with combinations of deep learning, convolutional networks, random forest, you name it, it was in there, and combinations thereof. Yet this simple autocorrelation measure with understanding of the physiology and the biology was superior to these techniques, which is really insightful. Okay, so I've just got maybe another 10 minutes, if that's right. Yep. Just to get moving to the last phase. So the NeuroVista trial ended, what was the mark, about 6 years ago? 6 years ago, yeah. And so it was unfortunate that that trial and that device doesn't exist today, because there was a lot of money and a lot of effort gone into the development of the device. And as we've seen from the data here, that the device actually works. The technology works. We can now show that we can forecast seizures, which is really amazing. But one of the reasons why it doesn't exist, because it was difficult to raise money to keep the trial going. It's very expensive. And that's because it's a class 3 medical device, which means that it's potentially life-threatening. It didn't have any therapeutic component. So there's risks for investors. But also the time it took for us to get to these results today, from when the data recording finished 6 years ago, was way too long. Okay, so what would we do different? Well, we'd move to a less invasive style device, and also we'd try and fix this data pipeline to speed things up. And of course, with all the insights that we've gathered, we'd do it ourselves. We wouldn't work with this other company, which was based in Seattle. So I'm going to talk about that for a moment. So Mark's leading a new company now, a new team, that's moving away from this style invasive device here to this less invasive device, which is a new company called Epiminder. Now this is a class 2 device here, so it's a very different game in terms of the regulatory burden, in terms of getting this through clinical trials. But from what we've learnt from the seizure forecasting from the invasive device, we're very confident that we can get the signals we need to better manage epilepsy from this style of system. So we're moving forward with this, and we expect the first in-human implant to be done later this year. So this is really exciting. But I'm really going to focus here on SEER. So I'm the CEO and co-founder with Mark, and Georgina Kenley is back in Melbourne at SEER Medical. And SEER was founded really to close this data pipeline. So to go from devices in patients that can take information around their behaviour, physiology and environment, get it up to the cloud, to be able to use data analytics and machine learning, and then get actionable feedback to the patient and to the clinic in a very fast way. And there's heaps of challenges involved in getting there. And so what we've really been working on is developing apps here. And so we have this product called the Beagle Seizure Tracking app, and Pip's been leading the development of this. We've got a very large team of people in Melbourne working on this cloud infrastructure here that can take in data and curate data. So it's a very exciting time there, because we can now use our current trial here and get the data pipeline closed, so we can get to from data to insight in a much faster way, which is going to give us a greater chance of success with this next generation of devices. And of course, we hope that SEER can work with Minder together to then accelerate the development of this new subscalp system as well. Now, the challenges we've had at SEER is that it takes years to go from device through to sales. And so what we've done with the infrastructure that we've been building is to start our own clinic. And so what we've been doing is running ambulatory video EEG. And so what we can do then is take patients out of the hospital, and we can run EEG diagnostic services from their own home and use our infrastructure to support the data analysis and diagnostic services. So we take referrals from neurologists, and the patients come into our clinics, and then they go home again for monitoring over a period of a week. And then we use our data pipeline to turn that data into clinical insight that can provide a diagnosis or characterizations of patients' epilepsy. So in about 2 short years now, it's gone very well for us. And so we've got 4 permanent clinics, 4 permanent offices around Australia, and we have 12 other remote clinics. So we service quite a large area. We've grown extremely fast to around 50 team members, most of which are in engineering. But we work very, very closely with our clinical team to iterate very, very fast on solving the problems that we face. And we're now serving around about 40 patients a week, which doesn't sound like a lot, but just to put this in context, most tertiary hospitals of serving these types of patients can service 2 patients a week. And that's because of the large demands with the data pipelines. So in fact, we're already looking after more epilepsy patients than the rest of Australia combined, because we use these excellent digital health techniques. And we service this huge area. So it's really making a big difference in Australia, where patients that wouldn't receive gold standard diagnostic care can now get it through our service. So just finally, just to wrap this all up, there's a really big opportunity right now in digital health space to move these ideas forward. And really what this is going to be driven by is these new technologies. So 5G networks are coming on board soon. So we'll be able to acquire data out in the field from patients in new ways that we never thought was possible. So that's opening up. Machine learning is really becoming democratized. So we use open source machine learning tools that have been developed by Google, like TensorFlow and others. And that's really just 10 lines of code. But really the big problems is how to organize and curate data in a sensible way where you can turn this data, which is the oil of the machine, into clinical insight. And that's really where our web platform comes into play. And so our clinical team, which is relatively large now, every click, every bit of information they put into the web platform is now used in curating data so the machine learning algorithms can get better. It's a very similar approach to what Google uses. Every click makes search better. So we want to try and reproduce this for the health system. And so we believe that this is going to lead to a new type of healthcare, new type of personalised healthcare, I should say, where we can distribute these types of monitoring devices in the home, where we can have a personalised real-time system. And epilepsy is a really great starting point for this, because we have to measure this broad range of physiological signals from behaviour to environment and physiology. So we're really excited about the potential for moving this forward. And so with that, I'll thank you and leave you with these topics for discussion. No worries. Thank you.
Any questions?
Are there any big pharmaceutical companies using Sphere? Drug companies?
Yes and no. So we have had research partnerships. There's one ongoing with UCB, and there's that's around. They've supported some of the development around our app because we provide this to patients for free, and it's very challenging to support that development. Now it's very interesting. So we thought that UCB would want access to the data, but in fact that's impossible, because they can't actually see whether or not a patient's taking their drugs or not, because if they don't take their drugs, it's considered as an adverse event. And they can't see if a patient's had a seizure, because that's considered an adverse event, and they need to report that to the authorities. So they're really doing this as just to support us, because they see the general good. But we've also used the at-home monitoring systems to better characterise patients' epilepsies, so to get a more accurate seizure count, because patients' own diaries aren't particularly accurate. So that's a really good tool for characterising epilepsy.
Have you considered moving away from implantable electrode technology towards magnetic field stimulation and detection?
Towards magnetic field.
Yeah.
I mean,
so some of the deep brain stimulation techniques, for example, have been using inductive approaches to stimulate the brain. I know they're not focused. I guess that's part of the answer as to why you're not using it. But
yes.
Of course, there's a huge advantage from the patient's point of view that you don't need to implant electric.
I think there's tremendous opportunities there. Really what we've focused on is techniques that we can get out as fast as possible, really. So the work with Medtronic, we're using really pretty old technology with the neural stimulators. And the work that Mark's leading with Minder is actually bootstrapping off the cochlear implant, which of course was developed in Australia. So that gives us an opportunity to move really from the bench to the clinic very fast, relative to a lot of other companies. And so we're focused on that, and also the form factors of these different devices. We need patients to have these for years or months, and I don't know if there's any magnetic stimulators that would fit the bill. Perhaps there may be. I'd love to hear about it. Yeah.
Have you thought about trying to predict arrhythmias in the heart? Because the exact opposite, you've got regular electrical activity in the heart, and it turns into a chaotic system as opposed to the normal chaos in the brain.
Yeah.
Into a
Yep.
organised seizure. Have you thought about, and ECG is obviously easy to monitor as well.
Yes, yeah. So we actually have to monitor ECG with all of our patients. And so there's actually research collaborators that we're working with to get the identified data from us. This is one of the great things about what we've been able to do with SEER, is to be able to acquire these really large volumes, 10 times the volume of any hospital of amount of data. And, it's a large team of engineers, so we feel like we're doing it right. And also the data goes up to the cloud, and it's curated, so it's accessible anywhere in the world. And so these other researchers are starting to tap into those data sources and investigating exactly these questions.
Are there any side effects to stimulating the hippocampus? Memory.
Yeah, look, it's really hard to tell at this stage. There's been a lot of work done, and there has been side effects shown in some patients, which they can show sleep disturbances. I'm not so sure about memory problems, but certainly there is side effects. But they seem to be rare cases, and what they're trying to do really is offset the effects of seizures. And seizures really have a drastic effect on memory. Often patients get their entire hippocampus removed, and their memory improves because they're no longer having seizures. I think Mark can probably talk about this a lot better than me, but
You're doing all right.
yeah. So yes, there is side effects, but the effects of the seizures are way more drastic. Yeah.
Right at the end, you were raising great hopes for artificial intelligence and machine learning.
Yes.
But a few slides earlier,
Mhm.
you showed that the simple methods you were developing outperformed
Yes.
all those complex. That seemed to be a bit of a contradiction in what you were saying.
Yes. It's really interesting. So I guess my message is, if you can always build a model where you can constrain your parameters by a fundamental understanding of the physiology, you're probably going to do better. But there is some problems where we can't do that quite yet. And so in SEER, for example, our big challenge is to get this analytics part working as well as a human would do. And so in a sense here, what we're doing is using labelled data from a human and getting, it's a supervised machine learning. So just trying to get that reproduction down. And we believe that it's possible with sufficient data. No one's done it well yet across a broad sense, but we certainly believe it's possible. But if we can find ways of constraining that, then we're going to go for it. But I mean, these tools now are, we use Amazon Web Services infrastructure, and the tools there are just so easy to use. It really has been democratized. It's, as long as you can organize your data and you have the right infrastructure, it becomes very easy to use these black box approaches. However, if you can, constrain the models, then you definitely should do it.
Maybe that's a way to close with an open question. I think it's probably one of the things that is going to be re-emerging, right?
Yeah, for sure. I think
the balance between black box and model-based and what you get from both sides.
yeah.
Yes.
Yeah.
Some people refer to it as giving chainsaws to babies rather than democratization.
Well, yeah, that's right. There is a danger zone. There's a big time danger zone. And so we have to make sure that our data is curated by the best, the experts. And that's, the data curation then becomes the problem. And getting that bit right, you can really make good advances.
I guess there is an open areas for mathematicians to think about how to constrain models, for example, other things. Nevertheless, thank you very much, Dina.
Yeah, thank you.